All right, let's dive in. Today, we're talking about one of the most revolutionary technologies of our time, gene editing. And look, this isn't some far-off science fiction anymore. It is actively reshaping our world, from the food we eat to the future of our health. So let's get right into it. To get started, just imagine this for a second. What if we could find and fix a single spelling mistake in the massive, complicated instruction manual that makes you, well, you? That, in a nutshell, is the incredible promise behind gene editing. It's about having the power to correct the very code of life itself. But, before we can really get into the editing part, we first have to understand the book itself. So let's start with the absolute fundamentals of our own genetic code. To make sense of this, let's stick with this book of life analogy. Think of your DNA as this incredibly detailed book. It's packed into pretty much every single cell in your body, and it contains all the instructions needed to build and operate you, right from the ground up. Okay, so if DNA is the book, then the genes are like the individual sentences on each page. And every single sentence, every gene has a very specific job to do. One might say, make the protein for brown eyes, while another gives the instructions for building heart muscle. When all these sentences are written correctly, everything just works. So what happens when there's a typo in one of those sentences? A mistake in the code? Well, that's where gene editing comes in. You can honestly think of it like a biological find and replace function you'd use in a document. It gives scientists this incredible ability to go into the DNA, find a faulty gene, and change it with pinpoint accuracy. Now, for the longest time, editing genes this precisely was just a dream. It was pure science fiction. But then a revolutionary tool came along that made it a reality, and that tool is called CRISPR-Cas9. This system basically has two key parts. First up, you've got CRISPR. Think of it as a super accurate GPS that can navigate the huge landscape of your DNA to find one single specific location, one specific gene. And then you have Cas9, which is like a tiny pair of molecular scissors that CRISPR carries with it. So how do these two work together? Well, it's pretty much a four-step process. First, scientists design a guide RNA that perfectly matches the faulty gene they want to target. Second, the CRISPR GPS uses that guide to zero in on the exact spot. Third, the Cas9 scissors make a clean, precise cut in the DNA. And finally, and this is the cool part, the cell's own natural repair systems kick in to fix the break. This is the moment scientists can delete the mistake or even insert a new correct piece of code. Okay, understanding the how is fascinating for sure. But the real story here, the reason this matters so much, is the why. This technology isn't just a scientific curiosity, it's already starting to rewrite our world in some truly profound ways. In medicine, the potential is, well, it's staggering. We're already seeing amazing breakthroughs in treating genetic disorders like sickle cell anemia by literally correcting the one faulty gene that causes it. The ultimate vision? A future where we can maybe prevent inherited diseases before a person is even born. And it's also completely transforming agriculture. Imagine crops that are naturally resistant to pests, which could slash the need for chemical pesticides, or what about wheat that can actually thrive during a drought? Gene editing is helping us create a more resilient and nutritious food supply for a planet that definitely needs it. And the impact doesn't stop with plants, it extends to animals too. Scientists are using gene editing to make livestock, like cows, more resistant to common diseases. Now, this not only improves animal welfare, but it could also dramatically reduce our reliance on antibiotics in farming. But, and this is a huge but, with this kind of immense power comes immense responsibility. This technology forces us as a society to confront some of the most complex ethical questions humanity has ever faced. And this right here is the central question. Where do we draw the line? Editing the genes of one person to cure their disease is one thing, but should we be making changes to human embryos? Changes that would be permanent, passed down through all future generations, forever? The concerns are really significant, and they are wide-ranging. There's the fear of heading down a path toward designer babies, where only the wealthy can afford genetic enhancements. There are the very real scientific risks of unintended off-target effects in the DNA. And of course, there are huge questions about social equity and the long-term impact on our planet's ecosystems. These are not easy questions, and they absolutely demand a global conversation. So, after all that, what are the key things to take away? First, gene editing, especially with a tool like CRISPR, gives us that precise find and replace function for DNA. Second, this is unlocking enormous potential across medicine, agriculture, and more. But finally, and maybe most importantly, this power confronts us with profound ethical challenges that we have to navigate carefully and together.
Maybe the best way to think about all this in our modern world is like this. If your DNA is the complex source code that runs the program of you, then gene editing is the debugger. It's a tool that lets us go in and fix the bugs in the code.
And that really leaves us with one last critical thought. For the very first time in human history, we hold the pen that can edit the book of life itself. So the ultimate question for all of us is this: with this incredible power, what kind of future will we choose to write?